Hi, good morning, everyone. I'm Hui Ling, your host. Welcome to today's webinar on data management tools for researchers presented by Dr. Christina Cho. Before we get started, I'd like to go over a few items so you know how to participate in today's event. The webinar is recorded and you can watch again after the session. You can submit your questions to Dr. Cho by typing them in the questions pane of the control panel. You may also send in your questions at any time during the presentation. We will collect these and address them during the Q&A. There will be a certificate that will be issued to all participants after the webinar. Please keep a lookout for the post-webinar email, which will be sent out in the next few days. Today, we have the pleasure of having Dr. Cho with us. I would now like to give a quick introduction about her. Dr. Cho is a cancer biologist who studies the role of the tumor microenvironment in the development and progression of solid tumors, including colorectal, pancreatic, and lung cancers. She is currently investigating the mechanisms underlying tumor-mediated immune evasion in melanoma and lung cancer. Without further ado, I'll hand it over to Dr. Cho for her presentation.
Thank you, Hui Ling. Hi, everybody. My name is Christina Cho. As I was introduced, I'm a cancer biologist. I'm currently a postdoc at Yale University. And I am here to talk to you guys about data management. And so I won't wait too long to get right into it. If you guys have any questions, please submit it into the chat. You can also follow me on Twitter and Instagram, as well as on LinkedIn. So if there are questions that I can't get to during this webinar, you can always contact me through those channels, and I'll try my best to answer your questions. So let's get started. So just a couple of, we're gonna go over a few topics today. So the main four topics are an overview of research data management, so a couple of definitions and the importance of good data management, and the best practices. So how you can go about doing research data management. We'll talk about the data lifecycle and how within the data lifecycle there are different stages, and for each of those stages there are specific techniques and tips that you can use to help manage your data in that stage. And then we'll go through a data management plan. Specifically, I'll do an example from the National Science Foundation, which has its own specific guidelines as to what they look for in a data management plan. And at the end, I'll be providing a list of websites and free resources that you can use to start managing your data. Now, the main objective of this webinar is so that I can provide you with some of the basics of research data management. It's not going to be all-encompassing, and it might not be as detailed as you'd expect because there's a lot of information out there, and there's a lot of field-specific information out there. And so if you have any questions that are very, very field-specific, I might not have all the answers, but I will try my best to direct you into the right source so that you have the information that you need. Hopefully you will walk away from this webinar understanding what data management actually is, and also the resources that you have available to you for free online. Okay, all right. So I want you guys to take a moment here and look at this cartoon. And I want to open up the chat to you. I want you to tell me what you think this cartoon is saying. Give you guys a couple of moments to write it in if you'd like. Oh, sorry. Okay. So this cartoon is a really nice image of how quickly data can become messy and disorganized and hard to dig through. That's a good analogy here, right? So the way you want to think about it is projects can last years, sometimes decades. I know for the field of immunotherapy, just from the discovery of the PD pathway to the actual use of the anti-PD antibodies was almost 20 years. So because projects can last years, there's a lot of change that happens. Locations of the labs can change. The people working on the project can change. For example, you might start on a project and leave the lab and someone else has to take over, or the opposite can happen. You might take over a project that someone else has left behind. But in that time, all of that data that's collected becomes this crazy mess if it's not well documented and not well organized and not well managed. So I want you to take this time to just think about your own personal experiences, right? Maybe you joined a lab and you took over a project and you went to go look for data and you couldn't find it. Or maybe you did find data, but it wasn't labeled properly, or you didn't understand how they got that data in the first place. All of that is part of data management. And another thing you want to think about is, what if you left the lab tomorrow? Do you think someone can walk in and take over and immediately pick up where you left off? Those are important questions to ask, because that is essentially why we need good data management. Without proper data management, data can become unusable. Not only will it be impossible to find important data, it won't be easy to understand what it means and how it was obtained. So what is research data management? We'll go over the definition as well as its importance. So I'm going to open up a poll to you right now. I want you to answer this question. What is research data? We'll give you about 30 seconds to a minute to answer this question with the multiple choice poll. All right, did you guys have a chance to fill out your poll? You have maybe a few more seconds left.
Hi, Christina. I'm actually showing the poll results now. So
Great.
let me read the results to you.
Sure.
So among the options, the most that people selected were actually statistical data. 55% of them selected that, followed by statistical records, then paper lab notebooks, then video and audio recordings, and software and codes.
Okay. So I'm sorry I did this to you, but this was a trick question. It's actually all of the above. So research data is essentially everything that is part of a project. So it includes statistical records, video and audio recordings, images, measurements, software and code, algorithms, and paper lab notebooks. And this is probably the most surprising for a lot of people, but certain biospecimens is also considered research data. So imagine all of this is research data, and all of this is something you have to somehow manage and organize. And that's what we're going to talk about a little bit today, how we organize all of this data. So what is research data management? Research data management, by definition, is the organization, storage, preservation, and sharing of data collected and used in a research project. It is the everyday management of research data during the lifetime of a project. So it's literally what you write into your notebooks. It's how you record your data. It's every time you get a new piece of information for your project, you're managing it as you collect it. Then there's also decisions about how that data will be preserved and shared once that project is completed. And all of these things are included into research data management. And what is the importance of research data management? I talked about it really early on with that cartoon. Well, one of the biggest things about research data management is that it verifies the integrity of your data. Data, especially digital data, it's quite fragile and easily lost. And so what good data management does is it helps to prevent errors in that digital data and also the quality of that data. And eventually, because the integrity is good, your analysis is more verifiable and reliable. Research management also makes your data findable and reusable. So as I mentioned earlier, sometimes you collect data and you might not use it. Someone else might use it later. They need to find that data again in order for them to use it. So you want to have a clear record of how that data was collected and where they can find that data. It also helps others understand your data. So there's an importance in context. There needs to be a context to what that data means. You can't just have a whole bunch of numbers on an Excel sheet if there's no information as to what those numbers mean. And also encourages other researchers to reuse and cite your data. The easier it is for people to find your data and the easier it is to understand it, the more likely they will reuse it and cite your data. So think of it like publicly available transcriptomic or proteomic data. If you know where the samples came from, you're more likely to use it because you can relate it to your own project. And lastly, and more importantly, more and more funding agencies are requiring research data management plans. And a lot of institutions now hire specialists. Librarians are trained for research data management at a lot of high research institutions. So it is very important now that scientists have some level of understanding of what research management is in order for them to get funding. So let's go into the components of data management. And we're going to start with what is called the data life cycle. So the data life cycle starts with a plan. The plan is a description of the data that you will be collecting and how that data will be managed and made accessible throughout its lifetime. Then the next stage is the discovery stage. You want to think about the potentially useful data, where it's located and how it's obtained, along with the relevant information about the data or metadata. So explanations and definitions and the importance of that data, like why you collected it and where it comes from. Then there's the actual collection and organization stage. So any observations that are made either by hand or with sensors or other instruments, and the data that's then collected into a digital form, that also needs to be organized. The next stage is the quality, so quality control and quality assurance. So the quality of the data has to be assured through different checks and inspections, and then controlled by describing those checks and inspections. How are you making sure that people can be confident that your data is of high quality? Then the next stage is the describe stage, or data description. You want to make sure that your data are accurately and thoroughly described using appropriate metadata standards. And we'll go through that a little bit later. So don't get too, hung up on trying to write all of this down. I will go through each stage and we'll talk about the best practices in each stage, okay? Then the next stage is storage, or storing your data. A lot of times data are submitted to an appropriate long-time archive, like a data center or repository, and it's really important that you follow guidelines for that. And the very last stage of the data is the sharing stage. And these are the questions you want to ask during this stage is who will have access and will this data be public? So for each stage of this data lifecycle, there are tips that you can use to help manage the data. And for each stage, I'll be going over those tips and those best practices. So the very first stage is plan. So research communities, institutions, funding agencies typically have their own standard methods and approaches for managing and disseminating data. So at Yale, we have a data management program within the library, and the librarians actually help us design our data management plans and also teach us on how to do the data management using software that's readily made available to us. And so after this webinar, it'll be very good for you to go to your institution and find out whether or not there are already people available to you to help you create a data management plan. So during the planning stage, you need to consider a few things. The first thing actually is the policies. So every institution has unique policies regarding licensing or sharing arrangements that pertain to the use of pre-existing materials. There's also plans for retaining, licensing, sharing, and in some cases, embargoing, which is the limitation, limiting the use of data by a specific group of people or a specific country for a period of time. You also have different legal and ethical restrictions depending on whether your studies use human subjects and other sensitive data. So policies are one of the first things you want to consider when coming up with a data management plan. You also need to think about the type of data you will be collecting. Will you be collecting brand new data through experimentation, or will you be reusing existing data? Also, will you be collecting measurements or biospecimens, audio recordings? That also makes a difference. The other thing you want to think about is the format of the data. A video recording is going to be formatted differently from an image, which is going to be different from an Excel file. You also want to think about versions. When you think about versions, you might think, that it's more complicated than it actually is. All it is if you have a file, a Word document, and you make it on day 1, and on day 2, you open it up and you make changes. Instead of just saving that file as the same name as the original version, you create a version 2 so that you're tracking the changes over time. This is really important if you consider projects that span over years and different investigators collect the same type of data using the same experiment. And sometimes version tracking is really critical to make sure that you can go back to the original data set if you need to. You also need to think of a backup policy, so how and where active data will be stored. You want to come up with a consistent backup policy and implementation strategy that is shared with any of collaborators that you have. You also need to consider storage and archiving options and requirements. If you decide to do long-term storage in a publicly available repository, they might have specific formatting requirements that you have to follow. You also need to consider describing and labeling. What this means is describing the data. So this is the metadata. And labeling is really important in terms of files. You want to make sure you have consistent labeling and nomenclature for your files. And it might be helpful to create a data dictionary. And this is something I'll talk about towards the end, but a data dictionary is essentially a record of how data is described and labeled. Then you think about access and sharing. So again, you might use a repository. You might even use a personal or research group website. And those are all going to determine how you format and label your files as well. You also need to consider rights and permissions. I mentioned earlier, every institution has its own policy about what can be shared and what cannot be shared. Licensing and copyright is really important depending on who you're sharing that data with. Another thing you might not consider, but is really important in the planning stage, is assigning roles and responsibilities. Projects are usually collaborative. It's not a one-person circus. It's many people involved. So you need to know who is responsible for what part of your project and which part of that project are they managing. Roles may include people who are responsible for data collection. Maybe there's another group of people responsible for data entry. So let's say you're doing a clinical study and you have a student that's helping you with data entry. That person's going to have a different responsibility than the person that's getting the permission slips from the patients, right? So you have to consider, all the people that might be involved in your project and how they play a role in data collection. You also want to think about the budget of your project. That seems like it's not very important, but this is especially important if you're creating a plan for a funding agency. They will want to know how much money it will cost to, collect all that data and then store it and then share it. So this is really important to consider from the very beginning of a project and from the very beginning of data collection. So now let's go to the data management plan, or a DMP. If you are applying for a grant for your project, at least in the United States, there is a very, very big chance that a data management plan will be needed, will be required. So you have to submit it along with your proposal. And so a data management plan is essentially a document that describes how you will treat your data throughout a project and what happens with the data after the project ends. And as I mentioned earlier, some funding agencies require a data management plan. In the United States, this includes the NIH and the National Science Foundation. And so I'm going to go through the requirements and the standards for the DMP of the National Science Foundation. But what you want to know for any data management plan is that there are 4 specific things you need to address. You want to address the data type, so the type of data that's to be collected, the data format, so this includes not just the raw data, but also the metadata standards. You have to think about the data sharing plan and how you're going to be archiving and preserving the data. So these are things that are included in a data management plan. A really good resource to use is Data One. You can go to the Data One website and go through they have so many different resources, but this website will allow you to create your own data management plan using examples that are uploaded onto their site. Another really good tool is the DMP tool. This is what's used predominantly at Yale and a lot of other institutions in the United States. So I'm going to go through an example of the National Science Foundation DMP. So according to the National Science Foundation, the length of a data management plan should be no longer than 2 pages. Now, this will differ depending on the funding institution. So it's really critical that you go to their website of the funding institution that you're looking to apply for and find out what the requirements are. The components of the Nsf Dmp include the types of data produced, the data and metadata standards, data access and sharing, data reuse and redistribution, archiving and preservation. So let's go through each component relatively quickly because we have to go through the other stages as well. Just to note, this is a recorded webinar, so you can always go back and look at this presentation for anything that went over too quickly for you, okay? So the types of data produced, that just means all the data, samples, physical collections, software, curriculum materials, anything else that you've produced during the lifetime of a project. And when you come up with the information that goes into this component, there are a couple of questions you want to ask. You want to ask what type of data will be produced for your project? How will that data be created or captured? What software programs are you using to generate that data? How much data will be produced? How big your files will be and how many of them there will be? And whether or not you will be using existing data. And if you are, what is the source of that data? Then you have the data and metadata standards. Basically, the standards you will be using for formatting and content. And for this, there are specific questions as well. You want to ask how you will document your data and project. Will you be typing it up into a README file? Will you be having a separate notebook where you write all of those details in? That's really important. You want to ask what file formats you will be using in your project and why, how you will organize those files into directories, and the naming conventions you will use. So how you name your files will be really important. How often will you be changing your data or updating it? And if you have different versions, how will you track them? How will you label them? And also, what type of metadata do you need to have in order for someone else to fully understand your data? So if you archive your data and someone collects it, for example, you go to TCGA and you download transcriptomic data, the metadata that comes with it will tell you the assay that was used to collect the transcriptional or RNA-seq data. And then let's go into data access, sharing, reuse, and redistribution. So this is all the policy stuff I talked about earlier. So you need to write in policies for access and sharing, especially if there's any type of confidentiality or security issues that might be there, or intellectual property rights. you need to include that into your DMP, at least for the NSF. And also, if you have specific policies for reuse and redistribution, especially if you yourself aren't the owner of that data, maybe your institution's the owner of that data, you need to write all of that into your DMP as well. And these are the questions you would like to ask when you are preparing this component. Who is responsible for the managing and controlling of your data? When and where do you intend to publish or distribute that data? How will that data be made available? Will there be an embargo period? Are there any issues regarding privacy or restricted confidential sensitive data? So if you're using patient samples or patient information, there are going to be some privacy issues you have to think about. There's then the question of any institutional review board protocols that are important for your research. And again, is there any intellectual property issues? That all goes into the data access, sharing, reuse, and redistribution component. The very last part of the Nsf Dmp is the storage and preservation. So this is your plan for archiving data, your samples, and other research products, and for preservation to access them later. And not just for you, but for the public to access them. And the questions here is, what is your strategy for data storage and backup? What kind of data will be preserved for the long term? Are there specific extra steps that you need to prepare that data for preservation? Where and how will that data be preserved? And what are the procedures that have taken place in order for you to ensure preservation and backup? And also, how long will that data be kept after the project is completed? So I know these are a lot of questions to think about, and this is a lot of information that I went over quickly. But again, this webinar is recorded, and you can go back and look at these questions as you think about your DMP. You can also go to the National Science Foundation website, and they have the guidelines available in a PDF format as well. Okay, so now we're only on stage 2. So we're halfway there in terms of time. So we're gonna go through this a little bit quickly, but please don't hesitate to ask me to slow down using the chat space. So stage 2 is discover. And the things you want to consider here is locating existing data and how you cite that data. So when you think about discovery of data, it could be in the context of discovering new data via experimentation or finding data that already exists. For example, published transcriptomic data on TCGA or GEO, any of those sites that have multiplex or multiplex or proteomic, like those big data sites, you might be downloading data from those sites. In that case, you need to know how to locate those existing data as well as cite that data. So how do you locate existing data? The easiest thing to do is go to a data directory. So there are many different data directories that have, they're online directories that maintain a list of data sources and repositories across a wide range of different disciplines. There are general repositories. An example is Figshare. And these are repositories that maintain data from a wide range of subject areas and are not limited to a particular discipline. Then there are discipline-related depositories. A great example is Dryad for the life sciences. And these repositories focus on a particular subject area or discipline or a cluster of related disciplines within the broad categories of humanities, sciences, social sciences, and government. So each category will have their own discipline-related repository. DataCite is a really good resource. It's an international organization that helps researchers find, access, and use existing data. So please write this down, and if you are ever looking for some kind of existing data that'll help with your hypothesis or push your project forward, this is a really good website to use. And the next stage in discovering is citing your data. You always want to provide proper recognition, cite the data sets that you've used, and also follow the guidelines and follow standards. And so every repository has a recommended citation standard, so you want to look at that. But the basic elements are title, author, date, version, and a persistent identifier, something that lets people know the source of this existing data. Okay, now to stage 3. This is the collect and organize stage. And these are some things to consider. So you want to be able to find and reuse data easily, right? So there are best practices for that. You also want to think about how you format your files and how you name those files and how you keep a good record of the different versions of your data. So let's start with organizing your data. Once you've collected data, you want to use it and analyze it. But if you can't find your data, you won't be able to use it or analyze it. So when you want to find and reuse data, you will need to have a way to type it in somewhere, type a name somewhere, and get the data quickly. So it's really important that you come up with a naming system for all of your data files and keep that very consistent. You also want to consider the format that you use to store your data. So for every type of file, you want to be consistent in the format that you use to save it, so that later on, when you go to find your data, it's much easier to locate it. Lastly, if you're going to archive or share your data, you need to consider the versions of that data. Again, it's like what I mentioned before. You might have a file that you started in 2021, and then 10 years later, someone else is working on the exact same project, the exact same experiment, and they're collecting a new set of data, and it's a different version. And so that's really important to consider when it comes to organizing your data, so that it's easier to find it later. So the file name. Before you start your project, you want to decide on a naming convention for all of your files. And the best practices include giving files a meaningful name. What that means is, as soon as someone sees the file name, they should know what it means. And usually a good file name that is meaningful might include a combination of the type of equipment used, or the date, or the researcher's surname. It basically allows the finder to know exactly what they're looking at. You also want to keep names at a reasonable length. Some applications don't work well with long file names, and a maximum of 25 characters is a pretty good rule of thumb. You want to also avoid using spaces or blank spaces in a file name. Instead, you can use underscores and hyphens to separate elements within a name. And if you're going to include the date or a date as part of your file name, use the standard of year, month, and date so that your files get sorted in a chronological order. And if your file name includes a numerical component, you want to have leading zeros. So a good example is like 001, 002, 003. If you keep those leading zeros, your files will sort in a sequential order. Lastly, you want to avoid special characters like exclamation point, question mark, $sign, hashtag, because some of those end up really messing up your file later, and it's hard to collect later down the line or find later down the line. All right. And as I mentioned earlier, you want to consider your versions. Here's a couple of websites you can go to look at different filing naming conventions for files. They have pretty detailed information that you can use. So Stanford University Library, the University of Wisconsin, Purdue, and Cornell all have data management services, and their website has free information that you can use. Okay, file format. So when you format electronic data files for long-term preservation and ease, you want to make sure it is really easy for people, ease of sharing, you want to make it easy for people to find your data and open it up. And there are obviously many different types of formats for each type of file, right? So image files, there's JPEG, there's the PDF file, there's so many different files, but you want to choose one and stick with it. And a couple of things you want to consider at the very beginning of data collection is you need to ask yourself whether or not your data will be in a format that is specific for the software that will be used to access it. Certain software requires specific formats. So if you're going to use a software down the line that is very picky, you want to make sure that you formatted that file appropriately. Also, if you're going to be depositing your data in a repository at the end of your project, you also need to consider the specific guidelines or requirements with respect to file formatting in that repository. And lastly, you need to think about the types of features that could be lost or modified while your file is converted from one format to another. So those are things you want to think about when you decide on a file format. And again, here are a couple of resources that you can use to get a little bit more detailed information. Again, Stanford, Cornell, and Cambridge all have data management services, and they have really nice websites that detail out all of this information that I'm talking to you about today. So data versioning, I said this several times. But basically, data versioning is saving new copies of your files as you make changes so that you can go back and retrieve specific versions of your files later. This is a really good example. So you have the data file name _1.0, and that would be your original document. Then you make some minor revisions and you call that data file name _1.1. And then maybe you make a different document with substantial revisions and changes. In that case, you would change your version number so that people know it's a distinct document from 1.0, right? So this is just an example of how you would keep track of your versions. And I'm going to go through 3 other examples that are commonly used for data versioning. So this one is the end of file name. So here you go. You see image 1, version 1, image 1, version 2, and so on. This is great, but sometimes what can happen is version 10 might move ahead of version 1 just because your computer reads it that way. The better type of versioning style is the dated style. So you include the image name and the date when you obtained that file. And a third style is incorporating names or initials of your collaborators. So you have the name of the file that describes what it is, then the date in which that file was collected, and the name of the collaborator or the individual who collected that data. So these are really good ways to record the versions of your data. All right, so let's go to stage 4, data quality. A really important part of data management is to develop a quality assurance and quality control plan, or QA/QC plan. And so just as data checking and review are important components of data management, so is a step of documenting how those tasks were actually accomplished. So you want to create a plan for how to review the data before it is even collected or compiled. And this allows the researcher, you, to think systematically about the kinds of errors or conflicts and other data problems that you are likely to encounter while obtaining your data. So you're basically thinking about ways to ensure that your data is of good quality and good integrity. So when other people look at your data, they can trust what they're reading. So this is something that a lot of people mix up often, and it makes total sense because assurance and controls are very similar. So the difference between quality assurance and quality control is that assurance is basically process-oriented and focuses on defect prevention, whereas quality control is product-oriented and focuses on defect identification. So quality assurance is also defined as all the planned and systematic activities that are implemented to provide confidence that the data will fulfill requirements for quality. So it allows people who are working on the project and those who are collaborators, as well as your funding agencies, to be confident that your data is of high quality. Quality control is more of an inspection aspect of quality management. An alternative definition is the operational techniques and activities that are used to fulfill the requirements for quality assurance. So it's how you make sure the assurance of your quality assurance is actually there. So what is the importance of a QA/QC plan? So a quality assurance, quality control plan is needed so that others can understand how to best use the data and avoid potential mistakes that might occur due to use of poor quality data. It also explicitly documents how you check your data for errors and conflicts so that you can help resolve current and future questions about the data. And a QA/QC plan should include methods to deal with erroneous data, so that would be assurance, methods to identify erroneous data, that's control, and another part of control is methods to mark that erroneous data. So how you would flag it. So let me give you an example. The researcher may graph a list of particular observations and then look for outliers. Then they'll go back to the original data source and confirm suspicions about those outliers. So a really good example, at least for me, is a tumor growth curve in mice. So I might have 50 mice and I measure the tumor growth over time. And I go to graph the data and I find that in day 30, I see this huge range of numbers. And I have to go back and look at why the range is so big. And I find that there are 2 data points that are outside of the mean. They're in the outliers. And I will go back at my data and ask myself, why in these mice did the tumors grow so much larger than the rest of the mice on that day? And maybe in my notes there, I have, okay, this mouse is actually, sick for other reasons, wasn't eating very well, had other issues independent of that tumor growth, and that outlier is not representative of the phenotype. So I can remove that data, but I have to record that. At some point, I needed to make sure that I had a plan to consider what is an outlier and when it's okay to remove certain data based on how I've defined an outlier, how I've defined data that needs to be excluded from analysis, okay? Okay, and also it's really important to record the type of methods that you use. So you want to make sure that for any given project or any given experiment, you are obtaining data using similar techniques and processes and environments to ensure similar outcome between data sets. I'll give you a really funny example. So there was a time, I don't exactly remember what the experiment was, but there was a lab that kept getting this really interesting data, but they couldn't reproduce it. And when they went and found out what was happening, it was literally because one investigator in the group, what she was doing was from taking the sample from 4 degrees to her bench, she had somehow held it underneath her armpit before she moved over to her bench. And that change in temperature affected the readout. And so just one investigator was doing something really odd that completely threw out the results, right? So you want to make sure that you are recording how you've done each step. And it's best if the techniques are consistent and the environment in which you do those experiments are consistent. You also need to have some kind of mechanism to compare the data set. So again, think of it as an internal control for specific experiments or studies. And in the cases for analysis, for quality control and assurance, is you want to use automation, such as script or macros or standalone programs, so that if you're doing a lot of repetitive functions, you actually maintain it the same way. So a lot of people use ImageJ to quantify immunohistochemistry or immunofluorescence images. And you go through a series of steps to get to your final image, right? You remove background, maybe you just salt and pepper. But if you use a script or macro, you will do the exact same processes for every single image every single time, instead of you going through and clicking everything on your own. So it's really good to use some kind of automation to maintain quality assurance and quality control. So now let's go to data description. Some things you want to consider is the metadata and your data dictionary. So what are some components of data description? The data description describes the scientific context. Why was the data collected in the first place? What were your questions? What was the hypothesis? Then you want to include critical information, such as the date or the location, not just the metadata embedded within the file name, but other information that is important for someone else to come in and know the environment in which you've collected that data. Identifiers within data sets are really important. So you have a table with measurements of all sorts. Within that table, you want to have a location where you've explained what each column means and what that data is, okay? So a lot of times you'll see in a supplementary file, there's an Excel file with 10 sheets, and in the first sheet, there's basically metadata. It's information about what the rest of the sheets will contain. The last thing you really want to consider doing is creating a data dictionary. A data dictionary does not contain data. It just describes the contents of your data files. So the variables that you used, formats, units of measure, definitions of coded values or missing values. For example, you're collecting survival data. You're tracking patients over time, and maybe patient A doesn't show up for one of the measurements. So now you have a missing value and you code that a certain way. So people know that that patient is missing on that day, that information from that patient, okay? So those are things you would include in a data dictionary. I've been saying metadata a lot. Some of you might not be familiar with metadata, but metadata is simply data about data. It provides context. So if you have a conversation, context drives understanding. The same thing goes for data. So data without context is really hard to understand. You won't know why it came to exist, where it was collected, and why it's important for anyone to know. So metadata contains context, and it basically describes your research data. And what does metadata do? It makes your data easier to find. It increases the understanding and reusability of your data. It also makes your data and associated research verifiable. So people can go back and look at your metadata and know exactly what you did to obtain that specific result. And what to include in the metadata. Some general information, kind of like your data set title, the name and contact information of the investigators, the date or the date range of the data that was collected, the geographic location of the data collected, your data and file overview, so just a short description of each file and the date when the file was created. Methodological information, so description of the methods that were used for data collection and data processing, and maybe some data-specific information, so a variable list with full names and definitions of the column headings if you have tabular data, units of measurement. Really what you're doing with metadata is answering these questions. Who created the data? What the data file contains? When the data were generated? Where the data was generated? Why the data was generated? And how the data was generated. So that's what the metadata answers. And so this is really critical. Where can metadata be collected? It can be collected in lab notebooks. It can be in text README files. It could be within the data file. Again, in an Excel sheet, it can be the very first sheet within that file. It can also be in web forms or electronic data collection systems. And lastly, what is a data dictionary? So a data dictionary just describes all of the data that's stored in a data set or used by a database. It describes the data, but does not contain the data. And the components of a data dictionary is a list of all the files, the type of data that's included in your data set, the list of field and variable names, and the description of information contained in each field. So a good example is for clinical data sets that have a lot of different clinical parameters, overall survival, staging. So a lot of times there's acronyms and symbols. What you want to have in the data dictionary is what those acronyms and symbols might mean. So here are a couple of examples you can go and search to provide you with an idea of what a data dictionary can look like. So there's AG Data Commons, there's also the National Renewable Energy Laboratory, and the Protein Data Bank Exchange Data Dictionary. Okay, let's go to data storage. We're almost there. So things you want to consider when you think about data storage is the size of your data set, your computational requirements. Again, if your file is really large and you need high processing speeds, you need to think about that. You also want to consider backing up all of your data. It's essential that you always have backup copies. And lastly, security. If you are working with sensitive data, such as medical data or human subject data, you need to make sure that your storage system meets security standards. So there's a backup rule of 3. You always want to have 3 copies of your data set. You keep your original copy, you have your second local copy, and then you have a remote copy. I have had very bad experiences without backing up, where I've lost data and had to go back and redo the experiment. So I'm telling you, always have 3 backups, or 2 backups and an original. So data sharing is the very last stage of the data cycle. Things you want to consider about sharing data is the benefits to it, the locations in which you can deposit your data, and how to prepare your data files for sharing. So there are obviously a lot of benefits for data sharing. It helps promote new discoveries. Someone can look at your data and have a brand new idea and a hypothesis based on the data that you've shared. It enhances the impact of your work. The more you share your research, the more likely people will cite your work, and your studies will have greater impact in the field. It also supports the validation and replication of your work. So other people will go and try to replicate your work, and that ultimately supports to validate your studies. It also encourages collaboration. More people will find your data, and more people will want to work with you. It also increases public investment. People are more likely to funnel money into funding agencies if your data is publicly available. It also reduces redundancy. You don't want other people doing the exact same studies you're doing and wasting time and money. So some locations for data sharing is a disciplinary repository, a data journal. A really good example of a really good resource is this Data at mLibrary blog. It has a list of data journals, and it's basically journals that only contain like information about data. So it's not like your results, it's a specific journal where you can deposit data. You can also do it as a supplementary file for your manuscript into a journal. And you can also use web-based tools like a wiki or a group website or a blog. And for preparations for sharing, you want to make sure you're using consistent and meaningful file names. Your variables and abbreviations should be pretty self-explanatory. You want to remove redundant variables and labels, and apply anonymization as needed. So again, if you're using human subjects, you need to protect their privacy. All right, everybody. And last thing is checking your copyright and privacy permissions. So let's quickly summarize and open it up to questions. So basically, data management is the organization, storage, preservation, and sharing of data that's collected throughout a research project. And data management is critical for every stage in the data lifecycle. And the things to always remember is record and track, name your files well, store and back up your files, and always look at guidelines and requirements for repositories, your institutions. You want to know what the policies are before you even start planning your project. All right, here is some free data management software. I'll leave this slide up, and I will take any questions from the audience.
Thank you, Dr. Cho, for the kind sharing. So now we'll open up the floor for questions. So feel free to send in your questions to the question pane in the control panel. So let me start with the first question. Dr. Cho, would you tell us more on what is the purpose of data management?
What is the focus of data management?
The purpose.
Oh, the purpose of data management. So the main purpose is the ability for you and anyone else to basically find your data and then use it properly and analyze it well. So I showed you that cartoon really early on, right? It's very, very easy for you to collect a lot of data and then at the end, not really know what you're looking at, what it means, and how to properly analyze it. A really good example, I can give you a personal example. So I worked on a project like 3 or 4 years ago, and I did some bioinformatics. And the issue with big data that you collect from TCGA is that they're always changing and uploading and updating things. And so the data that you collect in like 2014 is not necessarily the exact same data as you'll collect in 2021. But what you do with data management, especially with metadata, is you would have information about, where you downloaded that data, what it means, and the steps that you took to analyze that data for the figures that you may have obtained. Without that kind of record keeping of data management, you wouldn't be able to go back, so 7 years or something, and look at that graph and then now put in your manuscript the methods of how you got that graph, because it's been 7 years, right? The data management allows you to track your own work. And at the end, when it comes to publication and data sharing, you can trace it and you know exactly what that data means and how you got it. So that's what data management is really for.
Yeah, that's good to know. So the next question that I can see from the chat is, why would budgeting be important at the planning stage?
That's a really good question. So that is most important for when you're submitting proposals to funding agencies. So whenever you submit a proposal to a funding agency, they will ask for a budget, like why you need the money that you're asking for. And when you create a data management plan and you start thinking about how you're going to collect your data, where are you going to deposit it? Where are you going to publish it in a manuscript? All of that has costs. And it's really important to know what those costs are. So when you do submit a proposal and they ask you, why do you need this money and how would you spend this money, you actually can explain exactly where that money is going to. So the budget is really more for data management plans that are required for funding agencies.
Okay, the next question that I have over here is, how long and how safe will you keep most of your data?
So it depends on who you are. So if you are a PI, you want to keep your data forever because you don't know when, where, and how data that you collected at any given time will be useful later. If you are a graduate student, you will be saving and retaining your data in a lab notebook, in a hard drive, whatever, but that data is not yours. You do not own that data. You would leave it in the lab, and then the lab will have, your PI will store it. Similar with postdocs, you don't really own your data as a postdoc, your mentor does, so you can't really take it with you, right? I mean, unless your PI lets you, take some data with you, but basically, you want to keep your data stored and preserved as long as you can if you want to stay in the field of science, because you never know when that data will become useful. If you change fields, maybe it doesn't matter. But if you're someone like, I've been in the cancer research field for about 13 years, and I still go back to data that I collected 10 years ago to kind of get some ideas sometimes. Like, what if I had done this differently? Or is there a clue here that I can apply for something else? So I always recommend keeping good records of your data and saving all your files as best as you can.
Okay, the next question I see here is, what if the data is handwritten? Like we collect sentences from language speaker. How do you organize that kind of data?
Can you say that one more time? Sorry, I didn't hear the last part.
So I was saying about the handwritten data that people, collect from language speaker. So in that case, how would you, organize that kind of data?
Handwritten data. So,
Correct.
so the thing with handwritten data is there's a couple of things. So there's fragility, right? So if it's handwritten data, the ink, the paper, all of that can only last so long. So obviously, if it's critical that you have data that is sensitive to environmental damages, you need to then take measures to preserve that. But the other thing you want to do is create electronic records of those kinds of paper files. So I'm not in a field where that would happen very often outside of lab notebooks. But essentially, lab notebooks are kept for a really long time. I mean, there are labs that have lab notebooks from 20, 30 years ago. And they used to have carbon copies and things like that. But now a lot of labs are moving into electronic files as well. And so I don't know if I answered your question well, but basically for anything, any kind of data, so biospecimens, handwritten forms, whatever can be destroyed by environmental factors, you want to properly preserve those samples and those pages as well.
Okay, I will just pick up one last question. And just a note to all the audience who are still on the line, I have actually uploaded the slides for today under the handout section in the control panel. So you feel free to download, yeah, a copy for yourself. So the last question for today, we talk about collecting, organizing, and managing data. So how about, discarding data? Once you don't need that data, is there any process that, you use for that?
Sorry, I just dry throat. I talked a lot. Okay. So for discarding data, that's a really good question. So that's a very good question. I think when it comes to discarding data, you probably want to also include that in your data management plan under quality assurance and control. Now, if you collect data, let's say you're doing, again, I can only use from my own experiences. So let's say you're doing a tumor growth study in mice, and you have your first cohort of mice, and you get all of this tumor growth data. But you find out later that in that animal facility, there was an infection. There was a ringworm infection, and all of the mice developed it. And all that data you obtained, it's crap. You can't rely on any of that data, right? Because you don't know if the tumors grew a certain way because of the infection. You don't know if the mice were suffering. You don't know which mice had the infection, which mice didn't. There's just way too many variables that you cannot control for. In that case, you would remove all of that data and discard it because you can never use it again. That kind of stuff you include into your quality control and quality assurance. Like you need to have a plan there that allows you to remove data that is not reliable.
Thank you for that. Would you like to add anything more before we wrap up?
I don't think so. So I did get some pre-submitted questions, and there were a lot of really great questions. There was a lot I couldn't answer because it's not in my expertise. So I just wanted to, share something. So this is going to be in the PowerPoint, so you can definitely go and check all these references out, these websites. Somebody asked about qualitative data management. So I don't really do this. Qualitative data as like interviews, things where there's people-to-people communication. So that's a whole other field of data management. And the paper I cite here is a really good, actually it's a book. It's a really good resource you can use. It's Qualitative Research Methods, a Data Collector's Field Guide. And that's a really good guide to use to help you with quality data management. The other question I got was about geospatial data. Again, I'm not an earth scientist. There's very little I know about that, but this was a recent publication that came out in 2020 that helps you manage geospatial data. And the last one is someone asked about systematic review or for literature review. So for that, you would use what they call a review matrix. It's basically a spreadsheet that allows you to organize key elements. And this is a really good example. So you have your author and your title, your journal, the year published, the purpose of that paper, and other information that you might need. And you would use this Excel sheet to keep a record of the publications and the literature that you've read, so that when you go to write your manuscript, you can easily go and find the paper that you used to, put information into your introduction or discussion. And this is a website from VCU that talks about literature review and how you could use a review matrix for that. So these were some common questions I got in the submitted questions, and I thought I would share that with you guys.
Thank you so much for that. And thank you, Dr. Cho, for taking time to share your experiences. And a big thank you, everyone, for attending today's webinar. I hope you find this information useful. Keep a lookout for Wali emails or more webinars like this. Have a good day and see you at our next webinar. Goodbye.